What day is it of the week? What day of the week is it today? Wednesday? Okay.
I only know really how that goes.
Great. Thank you guys all for coming out tonight on this sort of gloomy Wednesday evening to talk about corals and hear an update on our project, which is funded by the RISEMBID program from the European Union, which is looking at improving coral restoration through assisted evolution, or basically selection of individuals. So I'm going to give you a bit of a background about our restoration program, and then I'll jump right into what we have been doing over the last year and a half with this project. And just for reference, I'm Dr. Gretchen Gabati-Gringley, and I am the Director of Research and Education at CCMI. I've been at CCMI now for 3 years, which is crazy. It seems like it's been very brief time, and I think COVID too probably played into that. But we've done a lot in those last 3 years, and CCMI is actually based on Little Cayman, so this is our facility here that you can see. Little Cayman is a tiny island just east of Grand Cayman. It's only 10 miles long and 1 mile wide, and the population is estimated about 160 people. So our lab on Little Cayman sits on the north side, which you can see. We have a couple of boats there that we use for research and education purposes. And we have a mission at CCMI, which is to improve our understanding of ecosystem function and the resilience of coral reefs. And primarily we're looking at how corals can adapt and acclimatize to changing conditions, and particularly those associated with global climate change. We're really interested in how ecosystems are going to function under changing conditions, and potentially identifying areas of refuge where organisms may be able to survive under extreme conditions. And our goal at the moment is to become one of the premier or leading research facilities in the Caribbean investigating coral reef resilience. So why are we looking at coral reef resilience? Well, if you're familiar with the scientific literature about coral reefs in the Caribbean and globally, really, you would know that they have undergone drastic declines over the last several decades. So in the 1970s, early 1980s, coral cover around the Caribbean was about 80% cover. That means like if you looked on the floor, 80% of what you looked at was live coral. And over the last several decades, that has declined to roughly around 5% coral cover region-wide. So this is a devastating loss, and it's due to several things, but probably the main drivers that we have identified are global climate change-induced bleaching, and so that's the loss of the symbiont within the coral, which is where it gets most of its energy, and disease outbreaks. And so there are other things that have also caused decline, such as overfishing. Hurricanes, for example, have had impacts, but all of these working in tandem have led to really low coral cover Caribbean-wide. And so we've gone from seeing these reefs that have been dominated by major reef-building corals that you can see in the top to a reefscape that is relatively flat and has a lot of macroalgae. And as I said, the main drivers of this change are climate change, so increases in seawater temperature, and we know that this has been happening over time based on long-term environmental data, and also acknowledgement of bleaching events that are happening on a more frequent basis. So we saw the first global bleaching event occur in the mid-1980s, the mass bleaching event. We had another one in 1998 associated with an El Nino event that caused a global bleaching event. And then since then, we've had them in 2010, 2015, 2016, and they're becoming somewhat of an annual event at this point. And this is important because it results in death of the coral if the coral cannot recover that algal symbiont. So the coral is essentially starving during this period of high temperatures. And another major cause of coral loss has been disease. So there are several diseases that affect corals across the Caribbean, things like black band disease, which you can see here. It's got this really classic ring of disease that surrounds the coral. There are things like white plague and white band, which particularly affects the acroporids or the staghorn corals. And then there are new diseases that have been emerging in the last 5 to 10 years, such as stony coral tissue loss disease, that have been devastating on coral reefs in the Caribbean. In Little Cayman, however, we have this sort of gem of a reef that has not experienced this extreme loss. So at CCMI, we have been monitoring the coral reefs there since 1999 on an almost annual basis. And our long-term data is showing that the coral reefs in Little Cayman have remained stable at about 22 to 23% coral cover. So if you remember from an earlier slide, compared to a regional percent cover of 5%, a 22 to 23% coral cover is very high. And so this indicates that perhaps Little Cayman is this really stable ecosystem relative to the region and represents a healthy example of what a Caribbean coral reef could be in this day and age. However, we are noting changes in species compositions on that reef scale. So we're seeing things like these large Montastria corals or orbicelids over here that form these big, like school bus sized corals. Those are the ones that we're losing, and instead we're getting much smaller corals that don't create the same sort of reef structure. And so even though on a whole we have this healthy system, we're still seeing impacts of change that are causing a shift in our community. But the one coral that has been lost in Little Cayman and really everywhere in the Caribbean are the acroporid corals. So Acropora palmata and Acropora cervicornis, which are the elkhorn and the staghorn corals. Now from this graph over here, you can see that from the historical dominance that we can see from fossil records, these corals were, 80% of the corals on the reef were these particular species. And really in the shallow water, it was just fields of these corals. So what we now would consider like our hard pan would have been covered with this palmata coral. And they've been lost primarily to an outbreak of disease. So in the 1980s, there was a white band disease that hit the cervicornis, and it devastated it. So roughly 90% of the cervicornis colonies across the Caribbean died from this disease. And this has now made this particular coral listed as an endangered species. And so this is the coral that we are focusing on for restoration purposes, because not only is it an endangered species, but it has a very high growth rate, and it's shown to perform very well in a nursery or garden setting. So we're focusing on restoring these habitats that have gone from ecosystems that look like this to ecosystems that look like this. And although we probably will never get back to this stage, we're at least trying to restore some of the functions that that ecosystem previously would have given to the environment. So how do we do this? All right, so we're not the first people doing this. People have been working on coral restoration now for almost 20 years. And so we have actually had our nursery in Little Cayman since 2011, and we've been growing corals on these racks, which you can see here. So we make these racks out of PVC and fishing line, and then we hang coral fragments from the fishing line, and they grow. And they seem to do really well in this setting. So bringing them up off of the reef into the water column there, away from things that may be encroaching like algae or cyanobacteria, maybe some ciliates or other things that might cause disease. They're separated from that, and because of this, they do really well in our coral nursery. They're also exposed to really high flow, so they're able to eat all the little zooplankton that pass by them while they're just hanging there in the water column. And people are using a variety of approaches. So when I first started working in coral restoration in Florida, we were using a system something like this, where we would make little pucks with corals on them on concrete blocks and put them in the sand. And we've really moved now more to this hanging strategy because the corals just grow much more quickly. And there is a lot of success in growing in the nursery. Where we seem to have an issue is from putting them from the nursery back out onto this reef. So that final stage of actually restoring the reef. One thing we have been doing at CCMI is trying to understand the best way to structure our nursery, the best way to outplant to increase survival. So one of the first studies that was conducted at CCMI was looking at whether the coral nursery did better at a shallow site or a deeper site. So here we're looking at the lab, which is just on the north coast of Little Cayman, and you can swim directly out to the shallow site, which is in about 30 feet of water, and a little bit farther than that, in 60 feet of water, would be our deep site. And these scientists were looking at the growth and survival of corals at these different sites, and they actually found that if you look at the deep versus shallow site, they performed better at the deep site than they did at the shallow. And we also found higher rates of disease and slower growth on the shallow site compared to the deep site. So because of that, we have moved all of our corals into this deeper site. So now we focus only on the one nursery, which is in our deep site. We then moved on to looking at the best strategies for how to outplant these corals onto the reef to maximize success post their nursery life stage. And we have tried a variety of different things, a variety of different habitats of where to put them, but more recently we've started using these spider domes that we build with rebar. They're dipped in like an epoxy and then covered in sand so that they don't impact the coral when they're touching the live coral that we put on it. We put them out onto the reef, and we try and find places where they're not going to be directly over top of a live coral, for example, so they're not shading something else that already is alive. And then we take fragments from our nursery and we attach them just with zip ties directly to these coral frames. And then we monitor them over time for how they're doing in terms of growth, survival, disease outbreak, etc. I will say, to measure their growth, we actually measure the length of every single branch and even the little nubbins that stick out from the side. So anytime we go back out there, any new growth that's coming out, even if it's 1 centimeter, a little nubbin, it gets accounted for into the total growth of that colony. So what we found from this study was that 6 months after outplanting, we had 80% survival of those colonies on the domes. And when we went out even another year later, so in year 2, we had an average colony survival of 73%. And this is really great because previously when we were outplanting directly onto the reef itself, we didn't have anywhere near that survival rate. And this is actually a 10-fold increase in our survival when we started using these domes. I will say, however, that in some of the other places like Belize where they're doing coral restoration, the success rate is similar to that. So roughly 60 to 70% survival is about what people are seeing for about 18 months. There are very few studies that follow the corals past 18 months. What we also looked at was disease. So again, we're looking at 1 year after outplanting and then 2 years after outplanting. And we found that the prevalence of disease didn't really change after 1 or 2 years. So there's about 30 to 40% of the corals showed white band disease, which may indicate that there are some corals that just are resistant to getting the disease, because otherwise we would have found that all of them would have gotten disease after that 2-year time point. So some of these corals are resistant, and that begs the question of who's resistant and can we identify those in our nursery and prioritize those particular corals. And all of this is really important because when we're doing coral restoration projects, we're not just trying to grow corals on the reef. We're really trying to restore the function of that reef. And when I'm talking about function, I mean every other organism that's interacting with those corals that we're putting out onto the reef. So when you restore a coral community, you're benefiting things like increasing the complexity of the reef structure. This is providing places for organisms to live and to hide, things for them to eat. So you're increasing your food source, not only for the corals, but also for the fish that live in that environment, or things, little invertebrates like lobsters or snails or whatever. And then through these interactions, you can have positive additional roles. So for example, herbivorous fish that come and live on this restored site are eating the algae, and they're creating clean substrate that can then be suitable for new corals to settle onto it. So all of these things are sort of interconnected and creating this ecosystem that's really functioning in the way it's supposed to be functioning. And when we actually went out and looked at how our outplanting strategies were affecting fish populations, we did find a positive benefit. So we found that after the 2-year mark, that there was a significant increase in the number of fish at the sites that we had outplanted our corals to compared to prior to before outplanting. So we did like a before-after comparison. There was not a significant increase in biomass, but the density shows that there is this trend of more fish recruiting to this location where we have restored this complex ecosystem. And so the next step with coral restoration is now trying to select for individuals that we're putting into these new communities that have the best chance of survival. So we know the two main things that are going to risk their survival are increased temperature and disease outbreaks. Right? So we can then start to look at ways to select for corals that might have a better chance. So we can do things like stress experiments with their larvae or their babies to see which ones are going to survive a heat wave, for example. We can do the same thing with adults. We can stress them out in different temperatures and see which genotypes or which individuals do better under higher temperatures. We can look at their past performance. So did they have a disease and recover, or were they exposed to a disease but didn't get the disease, that sort of thing. We can look at where they came from. Now in our case, all of our corals came from corals of opportunity several years ago, so they've all been living in the same environment for the last 5-8 years. And we can look at genotypic differences. So for example, like the differences between me and you, we're a different genotype even though we're the same species, but we may respond differently because of our genetic makeup to environmental conditions. And then we can select for those individuals and try to use them in our restoration strategy. So that's really what this project is trying to do. We focused on a few things that we could actually ascertain. So the things that we're looking at are past performance. We know that we've had disease outbreaks in our coral nursery. So who are those individuals that did well and who are those that didn't do well? And then we did some adult stress experiments, which I'm going to talk to you about, how we looked at differences in how they respond to temperature. And we looked at genotypic differences. So we actually have sequenced all the corals in our nursery. We know who is who, and we can use that information then to guide our practices going forward. So all of those things in combination have led to this present project. Like I said, this project is funded by Resembid through the European Union, Expertise France, and the World Bank, and we're very thankful for those funders for this project. So we're using, in this experiment, we're selecting for individuals that are resilient to heat and to disease, and we're looking for variations among genotypes. And we're going to then test these variables in a common garden experiment. So we're going to do this combined effect test, and then we're going to use those corals that we identified as different performers, put them out onto the reef and see how they do. So one of the first things we needed to know was their past experience. So who had been exposed to a disease, who got a disease, and who didn't get a disease. So like I said, we have all of our corals in the nursery have been sequenced for their genome, so we know genetically who they are. And we have them assigned just by letters, so G, Y, R, B, and K. This is work done by a PhD student, Anya Brown, when she was working at CCMI a few years ago in collaboration with several CCMI scientists. And what happened during her study was that in the nursery, we had racks that had only one genotype and racks that had mixed genotypes. And there was a disease outbreak that came through the nursery, and Anya was able to monitor which corals got the disease, which corals didn't, and then which corals recovered from the disease. And so this is their health status over time, and this is just across from June through October. So you can see that quite a lot of the corals did, in fact, succumb to this disease, but some of them, got the disease and then recovered from the disease. And when we look at that based on their genotype and whether they're in a mixed or single genotype frame, so in this graph, you have multiple genotypes on a frame is in purple, and a single genotype on a frame is in yellow. And so what you can see is if that yellow line comes up over the purple, it means that there was more disease in a single genotype frame than in a mixed genotype frame. And that is in fact what we saw for most of these genotypes. If they were alone on a frame, everything got the disease. If they were mixed in with different genotypes, then they were less prone to getting the disease. What we did find, however, was that one genotype in particular was very resistant to the disease, and this is genotype G, which I want you to remember because we'll come back to genotype G later in the talk. And then genotype K had the highest prevalence of disease, so almost 80% of those corals, regardless of whether they were in a single or a mixed genotype frame, got the disease. So this is a very vulnerable genotype. So we know their past experience, so now we need to look at how they tolerate stress. So to do this, we used what we call a thermal performance curve. So in the past, I have done this looking at really broad variations in habitat. So here I was looking at Bermuda versus Panama. So thinking that corals that experience really warm temperature all the time are really well adapted to hot temperatures, and corals that experience lower temperatures or maybe highly variable temperatures have a different temperature tolerance. And so you can make expectations or hypotheses about how your corals are going to perform. So here, for example, you've got the red lines are corals that would represent those coming from Panama, and the blue coming from the northern location in Bermuda. And if there is no adaptation to the climate in which you live in, you would expect those curves to completely overlap. But if they are adapted to those different conditions, then you expect them to start to diverge. So that's what we're looking for. And where you can use any metric for performance you want. It can be growth, it can be survival, it can be reproduction, and then you have a change in temperature, so you're testing different temperatures down here. For us, we're using metabolism, so we look at oxygen consumption and production, and we then can plot this. So this is gross photosynthesis, which is photosynthesis plus respiration. And again, you've got red is the hot location, which is Panama, and blue is the cold location, which is Bermuda. And you can see that they are diverging. They are different. So the corals from Panama did, in fact, have a higher temperature tolerance than those from Bermuda. So this indicates that this is a good way for us to assess differences in temperature tolerances. So we did the same thing here in Cayman with our corals from the nursery. So we took individuals of each genotype from the nursery, so we have replicates of each genotype. We just take a small fragment from our coral in the nursery and we bring it into the lab and we put it into these closed system chambers. Our chambers have a little spinning disc in the bottom, so there's circulation all the time, and then they have an oxygen probe and a temperature probe. We set this up in a cooler system like this, and it has heaters and chillers, and everything is controlled by a computer system on the top. So if it gets plus or minus 0.1 degrees C, the chiller will kick in and the heater will turn off, or vice versa. And so we can actually maintain our temperature at plus or minus 0.5 degrees C, which is great because we wanted to look at really fine scale variation in temperature tolerances. So we did this with coral with these different genotypes. And then we did it at a series of temperatures. I think we started from 27 degrees C and went all the way to 36 degrees C. So we're looking at all the different temperatures and how they're performing. Excuse me. All right, so what we found. So we've got the different genotypes here, and these are their performance curves, right? So you have your temperature on the X, and your photosynthesis, again, is on the Y. And these can be very confusing to look at. But what you want to look at are a few things like, at what point does this photosynthesis peak? At what point does it cross the zero, like it's dying? How steep is this slope? Those sort of things will imply whether a coral is vulnerable to temperature or tolerant of temperature. So first things, we're looking at that G genotype again. So this is the disease-resistant genotype. Okay, so this genotype actually had the highest optimal temperature, but it had the lowest thermal safety margin, whereas OB in orange had the opposite trend. So it had the lowest optimal temperature, but the highest safety margin. Safety margin is how much scope does that individual have once it passes its maximum temperature tolerance before it dies, right? So a very low safety margin means that once it gets up to that upper range, it doesn't have a lot of ability to tolerate much more. The other thing we wanted to look at was the slope of this line. So this is the excitation energy, EH. And again, G has the steepest slope of all of the different genotypes. Again, this coupled with that really small safety margin probably indicates that it doesn't have a lot of scope once it passes its optimal temperature to tolerate high temperatures, which may indicate that genotype G is actually less resistant to bleaching. So even though it's resilient to disease, it's not resistant to high temperatures. Whereas OB, on the other hand, might have a broader scope for tolerance for temperature. So you would expect that this would have a less risk of bleaching. And that is indicated again with G, which has the lowest maximum temperature that it can tolerate. Okay, so the next step with this project is we then took all of these individuals from our experiment. So fragments from the same colonies we used in our lab experiment, we then transplanted out onto our domes in the field. So we set up these domes here, just like the ones I showed you previously, and they all had representatives from each of our genotypes on each replicate dome site. And we measured them for total growth, so the length, and then we assessed a year later for survival, disease, and growth. So what we found is pretty cool. So we went back and looked at, for example, how much of the colony showed bleaching or paling. And that is represented in light blue in this graph. We noted if there were signs of disease, which is noted in dark blue. We then also, of course, assessed how much of the coral was healthy, which is green, if there was any evidence of partial mortality, or if the entire coral was dead or missing, as in it had fallen off of the dome. So again here, we've got these different genotypes on the bottom. And what I want to point out here, most importantly, again, is this genotype G. So we talked about it being disease resistant when there was a disease that came through the nursery. And now we're looking at it after we've outplanted it onto the reef, and none of the corals of genotype G showed signs of disease. So again, this is continuing to show that this particular genotype is probably resistant to white band disease. So that's really important in our future restoration prospects. But it also showed the highest prevalence of bleaching, which we probably could have predicted from those thermal performance curves. So that's also really exciting because it indicates that this method of trying to identify thermal tolerant or thermally vulnerable individuals can actually work. Interestingly also, genotype K, which you might remember is the one that had the highest prevalence of disease in the nursery when the disease came through with Anya's study, also had high prevalence of disease. K and KW have the highest prevalence of disease. So again, this is a vulnerable genotype to disease. However, neither one of those genotypes showed evidence of bleaching. So this indicates that there's probably a trade-off going on. So energetically, maybe they just can't tolerate both things. You can either be resistant to disease or you can be resistant to high temperatures, but you can't do both, right? So it's a win-win, win or lose situation. Who are you going to pick? And what this indicated to us was that our strategy going forward needed to be maximizing genetic diversity. We can't select for one trait because that one trait might then lead to it being vulnerable to another stressor. And so trying to maintain as many genotypes in our nursery and outplant into systems that have the maximum genetic diversity possible, we think is going to provide the best hope for survival of our outplanted coral reefs. And so that's what we did. So the last step of this project, we actually just did this in April over the Easter break. And my colleague from University of North Carolina came down and assisted with this. And we planted out 5 new sites around Little Cayman, and where we tried to do exactly that. So we took all the genotypes from the nursery and maximized the diversity on all of these 5 different sites. And we're going to continue to monitor those now over time to see if they do better than some of our previous outplants from the past, which did experience some disease. So the next step for this project is to go back in July, and we will do all of these metrics again. So we're going to remeasure all of their growth, their disease, bleaching, survival, all of that. And then we will have sort of the end solution, we think, to the best way to select for individuals to maximize survivals. And what we're going to do with all of this is we're going to write a restoration handbook for outplanting success, and we're going to share that locally It's going to contribute to the DOE's plan for restoration as well. And we hope that this will serve as a guidance for others who are doing restoration. Because as I said in the beginning, we're not the only people doing this. There are many people around the Caribbean and even globally working on coral restoration. And I think right now the focus of most of those groups is trying to really use data to improve our success. So not just going out and throwing corals on a reef and saying we did our job, actually really trying to make sure that we're doing this in the best way possible to ensure success of our reefs going forward. And with that, I would again like to thank our sponsors, not only the sponsors of this particular project, but also everyone who has contributed in the past and currently to our restoration efforts, which are ongoing and are extremely labor intensive. So we have to go out to that nursery every other week and make sure everything's clean. And if they're getting too big, we have to make sure we're keeping them trimmed and happy. And now we're also monitoring all of the corals that we have outplanted on the reef. So every sponsor is greatly appreciated, and thank you for your support. And thank you everyone for coming tonight. And I would love to take some questions. Yeah.
Are there any genotypes? I think you had 6 that you decided you weren't going to outplant, or you outplanted all 6? Or was it 7?
In the end?
Yeah.
So we outplanted all of them in the end. Yeah. And we actually added, there were a couple that we had in the nursery that we didn't have enough of them to use in the lab experiment for replicate purposes. And so we also included those in our final outplanting strategy as well. Yeah. Yes.
I'm curious on how we can volunteer, and like, well, in my case, I assisted in coral planting before, so I would love to get involved in some projects. So how can we?
Yeah, so here on Grand Cayman, there are a lot of options. So Ocean Frontiers, you can always assist with their work. I think, I'm not sure if Eco Divers is still doing stuff. No? Yeah, I would say Ocean Frontiers locally on Grand Cayman is the best way to get involved with restoration. Yeah. Yeah.
All of your genotypes then taken around little Cayman, is that correct?
Yeah, so initially when they were collected, they came from, I think, 4 different sites. And we've actually done a previous study where we looked at if we put them back at the site that they came from versus a different site, did it affect their success? And it didn't, so, which was kind of interesting.
And all staghorn, not so much elkhorn?
Yeah, we're not using elkhorn right now. Yeah. Although I think going forward, we are considering adding new species, especially now with skittledy that's raging. We want to make sure that we give every coral the best chance possible, because it's not just Acropora that's threatened at this point. Yeah. Do we have any online questions?
We don't.
Oh.
I've asked. Robin was saying the main reason that you chose the Acropora is because it grows so quickly. Were there any other reasons that one, I mean, aside from the fact that it used to be more prevalent than it is now?
Yeah, I mean, the main reason that restoration practitioners are focused on Acropora is because of that dramatic decline. So because it was really the first coral species listed as an endangered species, and also because it's really easy to propagate.
Yeah.
Yeah. Yes.
We have an online question
All right.
from Panchanuk. So the stage of coral lives affected coral performance. That is to say, larva and adult. I'd like to ask about the impacts of ages in the same stage, such as 1-year-old fragment and 3-year-old fragment. Have you tested this factor? So asking about like testing 1-year-old versus 3-year-old fragments.
So we actually can't tell the age of the coral when we take a fragment. So all of these corals have been in our nursery for quite a long time, and we just take a snip of that fragment. So, I mean, we can look at it in the sense that I snipped that fragment in 2020, and so that particular fragment I could say is 3 years old, but it could be from a colony that is 10 years old, right? So in that sense, we don't know, because we're not actually growing our corals in the nursery from larvae, although we are interested in doing some reproductive work and growing them from larval stages, but at this point we're just doing asexual fragmentation, so we don't actually know the age of each coral.
I have a question about the frame
Mm-hmm.
Do you need, I see that you have this mix of epoxy with sand, and with this, do you still need to clean the frames?
No, we don't. Yeah, we don't need to clean it. And in fact, the corals actually end up growing right over the rebar, which is great.
Okay, that's amazing.
Yeah. Yeah, this is one of our dome sites now. We just took this picture last week, so it's pretty good.
CCMI the first people to do this? Or?
With the domes?
Yeah.
No. This was developed, I believe, by the Mars Foundation. Started with designing these dome structures, and then we've sort of adapted it for our purposes. Yeah.
You had a flow chart earlier on with the branching of the staghorn corals as well. Some of them being more successful than others. Was that just a design on the chart, or have you noticed the branching of the corals being more opportunistic for the corals to survive better? Like you had 3 different ones in the flow chart earlier on.
Oh, you mean whether they're just one and whether they've got a branch?
Another, sorry.
Oh, so that figure, I think that's just to show diversity in potential structure. But we do find that the larger the fragment is, the better it does when we put it out onto the reef. So we do try and take large fragments off of the corals from the nursery rather than, like the size I'm using in the lab is like this big versus the size that I'm outplanting onto a dome, which is like that big. So, yeah. Yeah.
Have you noticed anything ecosystem-wise with the restoration, or are you going to start monitoring like which species maybe are showing up as getting some of the coral reintroduced, or just like what was lost, with the absence of it and things like that?
Yeah. So we're not looking at benthic stuff, but we did, before we put those 5 new sites out, we did fish surveys. So we did like a hovering like a roving diver survey, but basically you sit stationary and you look at an imaginary cylinder on top of where the dome is going to be, and we count all the fish that come in it in a certain time frame. And then we put the domes out and put the corals on, and then went out the next day and did the same thing. And then we did it at a site where we didn't put any domes as well. And then we'll do it again in July when we go back. So we'll have that before, after, and control for if the fish community is changing. Yeah.
So back on the side where you had multiple genotypes there, I think it's that one actually.
This one?
Looked like genotype OB looks a little bit interesting.
Yeah, so this one, again, showed no disease, but it did show some bleaching, which I find interesting because OB is the orange, and it had this really gradual slope and a really, the CT max didn't really change, the maximum temperature tolerance, but it had this highest thermal safety margin, so we would have thought it wouldn't bleach, but it did in fact show bleaching, but no disease. So I am also quite interested in OB. Like, I think that might be a winner in the long run as well. Yeah. I will say that my technician, Matt Doherty, made these graphs. Very nice. They took a lot of work. Great. Okay. Thank you guys so much. Appreciate coming out tonight. All right. Thanks.